Welcome to Lecture 6.1, in which we will provide an overview of cellular respiration before going into further detail in the next few videos. Our learning objectives for this video are to distinguish between autotrophs and heterotrophs and how they obtain nutrients, describe the overall reactants and products in the process of breaking down glucose by cellular respiration, understand the importance of ATP and NADH, and learn about kinases and dehydrogenases, two enzymes that play important roles in cellular respiration. As discussed previously, all energy comes from the sun, and producers are the organisms that harness this light energy and convert it into chemical energy. Producers are also autotrophs, which are organisms that can make their own food. Producers that specifically use light as a source for creating food in the form of carbohydrates are specifically referred to as photoautotrophs, with photo meaning light, like how we use light to do photography, auto meaning self, like how automatic doors are able to open by themselves, and troph meaning food. The word photoautotrophs means using light to make food for the self. Consumers that eat producers for energy are known as heterotrophs. Hetero means other, so the word means obtaining food from eating other organisms. The nutrients can then be converted into ATP, the energy currency of the cell, and the ATP can be exchanged to perform activities such as keeping our heart beating or our neurons firing. So how do we get ATP? Well, if we really want to simplify things, we eat food, which gives us glucose and other nutrients, and then we profit in the form of ATP. Glucose metabolism and cellular respiration is what actually connects the two. We take in glucose and breathe in oxygen, which is where the respiration part comes in. Our cells do the hard work of breaking the glucose down into carbon dioxide and water, and in the process, we get the ATP that we need. The mitochondria, of course, is essential to this process. You may have heard that the mitochondria is the powerhouse of the cell, and it is. It's how we're able to generate a lot of energy in the form of ATP from a single glucose molecule. Remember from the organelles lecture that the mitochondria has two membranes, an outer mitochondrial membrane and an inner mitochondrial membrane. The inner mitochondrial membrane is folded, which increases the surface area of the membrane, and these folded structures are referred to as cristae. The space in between the membranes is called the intermembrane space, while the innermost part of the mitochondria is called the matrix. Make sure to remember these different areas, because they will be relevant when we talk about where each step of cellular respiration takes place. Again, cellular respiration is the process of breaking glucose and other nutrients down to make ATP. And the oxygen we breathe is very important for allowing this process to occur. Overall, the reactants of the process are glucose and the oxygen we breathe in, and the products are the carbon dioxide that we breathe out and water. In the process, energy is released in the form of ATP. Ultimately, this is a process in which glucose becomes completely oxidized into carbon dioxide, which means that redox reactions must be occurring. One of the molecules that's critical for the oxidation of glucose is called NAD+. There are many redox reactions in cellular respiration, and many of these reactions involve reducing NAD+ into NADH, while glucose and subsequent molecules are oxidized. This is a structure which I do not want you to memorize. I do want you to recognize that NAD+ is actually made of two nucleotides, and that it's another good example of the function of nucleotides in energy. You can see the sugars and the nitrogenous bases here, and they're combined together by their phosphate groups. NAD+ is the oxidized form, and is reduced when it gets electrons and gains a hydrogen ion, also known as a proton. The small difference in structure that this makes is shown by the red circle. Since NADH now has extra electrons, it is also referred to as an electron carrier. FADH2, which is a reduced form of another nucleotide called FAD, is another important electron carrier. Both can be considered to be energy equivalents. They contain a lot of energy, the same way that ATP does, but cannot be used for energy the way ATP is. Instead, we will learn how the electron transport chain allows for NADH and FADH2 to transfer their energy to ATP. Speaking of ATP and NADH, you will find that the reactions that they're involved in often use the same types of enzymes. Kinases are enzymes that transfer a phosphate group from one molecule to another, and usually either uses ATP as a source of the phosphate group or creates ATP by giving it the phosphate group. The addition of a phosphate group to a molecule is called phosphorylation. This is a generic example. The substrate for a kinase must always have a hydroxyl group, and the kinase will essentially replace the hydroxyl group with a phosphate group, represented by this P in a yellow circle. Again, the phosphate group is commonly provided by ATP. The kinase is generally named for the substrate that it adds a phosphate group onto. However, kinases can also work in reverse. The same kinase that uses ATP to add a phosphate group onto the substrate can also take the phosphate group from this molecule and transfer it back to ATP. We will see one example of this when we go into glycolysis. A dehydrogenase is an enzyme that is involved in redox reactions, and specifically it removes hydrogens from one molecule, which oxidizes it, and transfers the hydrogen to another molecule, which reduces it. As mentioned, NAD+ is usually reduced to NADH in these processes, although there are other factors used as well. Like kinases, dehydrogenases are usually named for the molecule that they are dehydrogenating, that is, the molecule that they are removing the hydrogen from. Like kinases, they can also work in reverse, so the same dehydrogenase can also remove the hydrogen from NADH instead and give it back to the substrate. Now that we've learned a bit about the molecules and enzymes involved in cellular respiration, let's look at an overview. Cellular respiration is divided up into 4 phases. The first is glycolysis, with glyco meaning glucose and lysis meaning breakdown, so literally glucose breakdown. This creates a molecule called pyruvate, which then enters the mitochondria to undergo further oxidation. This creates acetyl-CoA, which enters the citric acid cycle. Throughout this process, ATP, carbon dioxide, and NADH and FADH2 are being produced. At the end, the NADH and FADH2 will go through the electron transport chain to generate even more ATP, as well as water. We will talk about each of these phases in more detail over the next few videos. In this video, we learned that autotrophs are organisms that are able to create their own nutrients, and heterotrophs eat other organisms to obtain nutrients. For both, these nutrients are broken down by cellular respiration to produce ATP. The overall inputs of cellular respiration are glucose and oxygen, and the outputs are carbon dioxide and water, with ATP created from the energy released during the process. ATP is the energy currency that allows for our cells to work, while NADH as well as FADH2 play a role in redox reactions during cellular respiration and are used to produce more ATP. Kinases are enzymes that can use ATP to phosphorylate molecules or remove phosphate groups from molecules to make ATP. Dehydrogenases will oxidize molecules by removing hydrogens, often producing NADH or FADH2 in the process. But like kinases, they can also perform the reverse reaction. In the next video, we will begin learning about glycolysis, the first step of cellular respiration. Thanks for watching.